Alright, so we're going to focus our attention on some other non-Mendelian topics. Alright, so we're going to look at pedigree analysis. Alright, so whenever you're asked to set up a pedigree, you're going to be looking at family histories, genetic conditions, who had what, how many offspring did they have, did they exhibit the same trait, did they or did they not. And so here we're looking at, for instance, attached versus free earlobes. So again, having free earlobes is a dominant trait. Having attached earlobes is a recessive trait. Now, how do you go about figuring out the genotypes of each of the individuals in a pedigree? Alright, so some of the information you're going to be given is whether or not the individuals are male or female. Right, whether they're symbolized by a square or a circle. Squares are going to be your males, and circles are females. Alright, so then you're looking at affected and unaffected. Alright, so in this case, looking at individuals that have attached earlobes, in this case affected, they're shaded. Individuals who are not affected, in this case, who have free earlobes, are not shaded. Well, one of the things I do is I set up all the possible combinations of Punnett squares, alright, for this given trait. All the possible crosses, whether the individual is homozygous dominant or heterozygous or homozygous recessive, and who they're mating with and what their genotype is, or could be, and the offspring in question. Alright, and this can give you an idea of whether or not it can give you an idea based on the crosses, the outcome, the genotypes of the parent. Based on, you're going to have to know about their offspring. Right? If they don't have offspring, you're going to be kind of left wondering what their genotype is in some instances, and we'll talk about that in a little bit. Alright, so you need to know their offspring and whether or not they're affected or unaffected. Alright, so that's one of the things you also have to know. Alright, so here, alright, I've set up all the different Punnett squares. Alright, so here we're looking at a simple monohybrid cross. Alright, looking at the inheritance of one characteristic. Alright, so earlobes, attached, unattached. Alright, so here we have two individuals that are homozygous dominant that have, in this case, free earlobes. Well, as should be expected, all their offspring have free earlobes. Right here. What if one of the individuals is heterozygous? Well, then it's going to be still all of your offspring have free earlobes. But some are homozygous dominant, some are heterozygous. Here, two heterozygous individuals, 1 to 1 genotypic ratio, 3 to 1 phenotypic ratio. So 75% of the offspring, or 75% chance that your offspring are have free versus attached earlobes. Heterozygous and homozygous recessive. Alright, so free earlobes crossed with an individual that has attached. Alright, two individuals that have attached earlobes are going to have all their offspring have attached earlobes. Okay, so let's look at, in this pedigree, individuals A and B. Alright, so by looking at the pedigree, alright, you see that individuals A and B, alright, grandpa and grandma, have both free earlobes. They're unshaded. Alright, so what are the possible genotypes for a person who has free earlobes? Well, they're either homozygous dominant or they're heterozygous. Well, which is it? Well, here are the possible combinations. Alright, now, looking at their offspring, alright, they have children that have a mixture of attached and free earlobes. Alright, so that means if all their offspring, you would expect if all their offspring had free earlobes, then the grandparents in this case would be either both homozygous dominant, or one would be homozygous dominant and one would be heterozygous. But that's not the case. Here you have a mixture. Alright, you have a mixture of individuals with free and attached earlobes. Alright, so that tells you that in this case, somebody is inheriting a recessive allele from both parents. So here, individuals A and B are going to be heterozygous. That's the only possible outcome for this particular situation. Alright, so what about individuals C and D? Well, here's a cool piece of information. If you see that individual C here is shaded, that tells you that individual C is affected. Well, if you're affected by this condition, in this case you have attached earlobes, then you automatically know the genotype of individual C. You know they're homozygous recessive. So you know that individual D is either homozygous dominant or heterozygous because they have free earlobes. But when you set up your cross, you know you're crossing a person who's homozygous dominant or heterozygous with an individual who is homozygous recessive. Alright, so now look at the offspring. Well, you have offspring that have free and attached earlobes. Well, if you expected all your offspring to have all free earlobes, that would mean that individual D would be homozygous dominant, which is not the case. We don't see that. Both of their daughters have either attached or free earlobes. In this case, that means individual D is heterozygous. Now, let's look at the result of our cross between individuals A and B. Alright, so they are both heterozygous, so we can have a prediction of the possible genotypes of their offspring. Alright, so we know that individuals F and G are affected. We know they have attached earlobes, they're shaded. So we know that they are homozygous recessive. Now, individuals E and H, we see that they have free earlobes. Well, are they homozygous dominant or are they heterozygous? How do we figure that out? Well, here, individual E. Individual E, we don't have offspring for this individual. Alright? So, since we don't have offspring, all we know is that she has free earlobes. So right now, we're going to designate her as either homozygous dominant or heterozygous. Now, individual H. Individual H is going to have offspring with another individual. And we see that their offspring are going to have free and or attached earlobes. Alright, so that tells me at least that individual H is contributing at least one attached earlobe allele. So that tells me that this individual, this male, has to be heterozygous. Alright, so looking at individuals I and J. Well, J is affected. Alright, he has attached earlobes, or she does, sorry. She has attached earlobes. So we know that she is homozygous recessive. Now we can look at the product of this cross between individuals C and D. You're either heterozygous or you're homozygous recessive. Well, that makes it easy for us to figure out I. Well, I obviously has to be heterozygous. Alright, well, the product of this cross, alright, two heterozygous individuals, we would get the offspring K and L. Well, here, individual K is affected. She has attached earlobes. She's homozygous recessive. Individual L, she has yet to have any offspring. So we don't know with confidence whether she is homozygous dominant or heterozygous. So we put down both for right now. Now, these next couple of slides are just showing you different types of pedigrees. Okay, I'm not going to have you look at a pedigree and tell me whether or not the traits that are being passed on from one generation to the next are autosomal or sex-linked, whether they're dominant or they're recessive. Right? You go on to take a genetics class, you'll do this. Now, you could potentially hypothesize, all right, and based on the knowledge you have so far, give a prediction. All right, this is just showing you some of the characteristics of pedigrees of individuals that suffer from autosomal recessive traits like sickle-cell disease, how this is passed on, what the pedigree would look like. All right, for an autosomal dominant trait like Huntington's disease, right, what the pedigree would look like. All right. Individual with a sex-linked recessive trait, like red-green color blindness, what that would look like. And then here, a sex-linked dominant trait, right, what that would look like. Right, but you can see here there's all different patterns of who's affected and who isn't, and probabilities of who and who is not affected. Alright, so looking at some other topics. Alright, so far it's been pretty straightforward. Alright, we talked about situations where we have complete dominance, meaning that in a heterozygous individual, alright, the phenotype of the homozygous dominant individual is expressed. Okay. In other words, the dominant allele is expressed over the recessive allele in a heterozygous individual. Okay. So here, we're looking at gerbils. I know, they're cute, they're adorable. So looking at their color gene, alright, so we have a Goody or brown allele, which is dominant, to the black allele. Alright, so an individual that is a Goody or brown, they can either be homozygous dominant, or they could be heterozygous. And if they're heterozygous, the dominant agouti allele is expressed, so they have brown fur. Alright, so this is similar to Mendel and his pea plants. Recall that the pea plants, the purple flowers, they have at least one copy of the purple allele. The enzyme involved in pigmentation for the purple coloration, pigment production, is expressed. Alright, so here it's the same thing. You have production of the brown pigmentation. Alright. Here, for the black allele, alright, you have another set of genes. The black allele, our little black gerbil, is homozygous recessive. Alright, so here he's expressing the black allele. So you have the black pigments that are produced. And this is only going to be expressed in a homozygous recessive individual. These guys here express the dominant phenotype. These guys here express the recessive phenotype. Alright, so this is complete dominance. Alright, so what about incomplete dominance? Alright, so thinking back to the very first topic we discussed in this chapter, we talked about this idea of a blending hypothesis, right? Well, incomplete dominance is an example of a situation that looks like the blending hypothesis might hold true, but in reality it doesn't. Alright, so here in incomplete dominance, you have a heterozygous individual, okay, and this heterozygous individual is going to express a phenotype that's an intermediate between the dominant and recessive phenotypes. All right, so in incomplete dominance, there's no dominant allele. Okay. All right, so here, for example, you have red alleles, you have white alleles. Okay, so here, a flower with red alleles is going to have this genotype. A flower with white alleles will have this genotype. And a flower with pink, a flower with, a pink flower will have a genotype that'll look like this. Have a copy of the red allele, have a copy of the white allele. Alright, so in this case, they have a reduced expression of this red allele. Alright, the reduced expression of this red allele leads to the development of pink flowers. Alright, because the red flowers have two copies, so they have double the expression of the red allele. The white flowers have no pigmentation. The pink flowers have reduced red colored pigmentation, so they're pink. Alright, so here we're looking at an example of incomplete dominance with our SNAPDRAGONS. Alright, so we take a homozygous red flower and cross it with a homozygous white flower. Alright, and we get all offspring that are pink. Alright, so they have a copy of the red and the white alleles. Okay, so they're all pink flowers. Well, what happens if we cross two of these pink flowers that are heterozygous? Well, you see the return of the parental phenotypes. So again, the blending hypothesis doesn't hold true because the parental phenotypes come back. Alright, so you see that you have flowers that are white, flowers that are pink, flowers that are red. Because both parents can contribute either a red or a white allele. Codominance. Codominance involves, again, another heterozygous individual that's going to express both alleles equally. Again, there's no dominant allele, but both alleles get expressed equally. Alright, this is opposed to incomplete dominance. Alright, so you see a reduced level of expression of genes. Codominance, you see an equal expression of genes. Now, an example of codominance is the AB blood type. Alright, so individuals who have type AB blood, alright, they have a copy of the A allele, they have a copy of the B allele on the surface of their red blood cells. Both alleles are expressed equally. They are codominant. Now, what about individual that has type A blood, but also has a copy of the O allele? Well, in this case, a copy of the O allele. Alright, the O allele is recessive to the A allele. Alright, so here you do have an example of complete dominance. The A allele is dominant over the I allele. Alright, what about individual that's type B? Individual is type B. Alright, the B allele is dominant over the O allele. Alright, again, complete dominance. But the A and the B allele, neither is dominant over the other. This is also a situation where we're dealing with multiple alleles. Alright, so far we've talked about red, white, black, white, albino, non-albino, dwarfism, no dwarfism. We talked about two different situations, basically two alleles. All right, here we're talking about multiple alleles. All right, we're talking about an A allele, a B allele, an I allele. All right, so here, individuals that are type A blood have either two copies of the A allele or a copy of the A allele and a copy of the I. Individuals who are type B blood have two copies of the B allele, a copy of the B allele, and the O allele. Individuals who are type O have two copies of the little I allele. And individuals who are type AB have type A allele and type B allele. Alright, so you'll need to know the different genotypes of an individual as type A, type B, type AB, and type O blood. Alright, so let's look at a little situation here. Alright, just for example, what if we have an individual with type A blood? And let's say we have an individual with type B blood. Alright, well, here, set up your little cross. Type A, type B, and the O allele. Do your little Punnett square. Alright, so we got AB, we got AB, we got A, and we got A. Alright, so the offspring of this cross would have either type AB or type A blood. Alright, so doing crosses with blood types is just like any other monohybrid cross. Alright, Pleiotropy. Alright, so this is where you have a single gene that affects multiple traits in an individual. An example would be Marfan syndrome. Alright, so fibrillin 1 gene. Alright, so in this case, the mutant allele is dominant over the normal allele. So this is a dominant inherited disorder. Alright, so that means to exhibit this condition, you either are homozygous dominant or you are heterozygous. Individuals who don't suffer from the condition are homozygous recessive. Alright, so here you have an individual with disproportionately long limbs, fingers, as well as vision and heart problems. Polygenic inheritance. Alright, so here you have many genes that affect a single trait. Alright, so here looking at human height. Alright, there are many genes that affect human height. We also see an array of individuals, spectrum of heights, going from really short to really tall to those that fall in between. So there's a nice spectrum. It has this nice little bell-shaped curve. You see the same thing with skin color. Alright, you see many genes that affect skin color. Alright, so you have individuals that are really light, individuals that are really dark, and those that fall in between. You get this nice little bell-shaped curve distribution. Epistasis. Epistasis, you have one gene that is masking the effects of another. Alright, so one of the common examples is the Labrador Retrievers. Alright, so we have three major varieties. We have Golden, we have Black, and we have Chocolate. Alright, so here you have either pigment color. You have a dominant black allele or a recessive brown allele. So that's a gene for pigment color. You have a separate gene that's involved in actually taking that pigment when it's produced and depositing it into the hair follicle. Alright, so here the dominant allele allows for this dark pigment to be deposited. The recessive allele prevents the pigment from being deposited in the hair follicle. Alright, so here a Labrador Retriever that has a black coat color. This individual can be homozygous dominant for pigment color and pigment depositing. They can be heterozygous for pigment color and homozygous dominant for pigment depositing. They can be heterozygous for both genes. They can be homozygous dominant for pigment color and heterozygous for pigment deposition. But either way, they have at least one copy of this dominant E allele, meaning that the dark pigments are going to be deposited in the fur coat. Now, a brown-colored lab, a chocolate lab, has the recessive alleles. So they have two copies of the recessive allele. They are either two copies of the recessive allele for pigment color. They have either two copies of the dominant allele for pigment depositing, or at least one copy where they're heterozygous. Either way, they still deposit the brown pigment color in the fur. Now, if you have an individual that has two copies of the recessive allele for pigment depositing, alright, so they're homozygous recessive for that gene, it doesn't matter what the genotype of the gene for pigment color is. It doesn't matter if it's homozygous dominant, heterozygous, or homozygous recessive. It doesn't matter if it was going to be a black or a chocolate lab. If you don't have at least one copy of the dominant E allele for pigment depositing, that pigment protein does not get deposited in the fur. So you end up with a lab that has a golden or yellow coat. Alright, so you have this pigment depositing gene that is masking the expression of pigment coloration in the dogs in terms of the fur coat. Alright, so here you're looking at a cross between two labs. Alright, so here you have two black labs, alright, that are carriers of the recessive pigment depositing allele. Alright, so here you end up with a mixture of offspring that are black labbed, chocolate labbed, and golden.